All right, well, this is our lecture for Monday, and this week on Monday we're going to address some big challenges presented to macroevolution, and some would argue that these are insurmountable challenges. And then on Wednesday, they take basically the exact opposite approach and argue that macroevolution is the most reasonable conclusion from the data. So let's deal with this first one. Is macroevolution even possible? And so, in order to address this, we need to talk about four potential hypotheses about how evolutionary change actually takes place. And we'll come back to these hypotheses towards the end of our lecture and see which one makes the most sense. So the first one is that universal common descent is true and it functions using Darwinian mechanisms. The next one is that universal common descent is true, but it uses some mechanism other than Darwinian mechanisms. The next one, this is an interventionist hypothesis. Universal common descent is not true, but microevolution works primarily by Darwinian mechanisms. And the last one, again, another interventionist perspective, universal common descent is not true, and even microevolution requires some more sophisticated mechanisms than the Darwinian mechanism. So as we go through these, as we go through these slides, I want you to keep these hypotheses in mind. So next thing, don't we have plenty of evidence for new biological features? And this is exactly what we mean by macroevolution. We mean the evolution of major new biological features that require new and very sophisticated genetic machinery. And so here's some examples. There are a number of crustacean species that have two different types of thoracic appendages. And what you find is that the two different types of thoracic appendages are developed because of the work of two different Hox genes, two different master control genes or regulatory genes. And so this provides evidence that originally you had one Hox gene, it was duplicated. The duplicated gene had the opportunity to evolve some new functions, and then as it works on the appendages, it actually develops them into different appendages. Humans have duplicated primate genes. The idea here is that primates, some genes in some lesser primates duplicated, and those duplicates had the opportunity to evolve and develop some new function, and with that, you were able to add to the complexity of these organisms. We see insects develop resistance to insecticides. We see bacteria develop resistance to antibiotics. Sorry, that should say antibiotics. And all of these on the surface seem to be evidence for new biological features. Now, what's interesting is the first two, these are assumed that this is what developed these organisms. In crustaceans, it could very easily be explained, just as easily be explained, that these two Hox genes always existed and they were designed to, from the beginning, generate these two different types of appendages. Again, with the second one, we could assume that humans were created separate and unique from other primates, and so those genes that are similar to primate genes that look like they were duplicated were there from the very beginning, and the similarities need to be explained in some other way. Now, the latter two, these are more difficult because these are things that we've actually seen take place. We haven't seen humans evolve from lesser primates. We haven't seen crustaceans develop, two different types of thoracic appendages from one. But we have seen insects develop resistance to insecticides, and we've seen bacteria develop resistance to antibiotics. But what you find when you actually study these at the genetic level, they're almost always a result of a loss of genetic information, and when they aren't, they're generated by the combination of genetic pieces that are already in place. So this is a question we've asked several times. Is macroevolution a reasonable extrapolation of microevolution? And so artificial selection experiments would seem to illustrate that it's not a reasonable extrapolation. Artificial selection, we're able to put some enormous selective pressure on these organisms, knowing exactly what we want and trying to make that happen as quickly as possible. And very quickly we see that there is a limit to the amount of variation that we can get from the present genetic information. So these limits to the morphological forms that we have are determined by the genetic potential. Now the proposal is if you could have millions of years of time, you could generate some new mutations that then we could bypass these limits. And so is macroevolution a reasonable extrapolation of microevolution? Well, the data suggest it is not, but the data don't, tell us that it is impossible. So we need to explore this further. So does natural selection generate new structures? And this, mutations are what generate the variation, but in terms of generating new structures, natural selection needs to select the mutations that add to the fitness of an organism, and over time the slow buildup of these mutations, these beneficial mutations, can generate new novel structures that increase the complexity of these organisms. And so here's the main idea, or not the maybe the main idea, but a main idea of how this takes place. And I've mentioned this a little bit already. The idea here is you have your original gene, you have some gene duplication event. You can see that here as well. Here's our original gene. Here's a gene duplication event. The original gene is still under the pressure of natural selection to function as it ought to. The other gene, though, its product is unnecessary. This original gene is already producing that protein product. So the idea here is that natural selection would allow this duplicated gene the freedom to change. Now, it could lose its function altogether, but it could maybe gain some new function as we start to accumulate these mutations in this new gene. The idea here is, it could become non-functional, but it may in fact actually develop some new function altogether and add to the complexity of these forms. So the real question here is we have to deal with is there evidence for duplicated DNA? And so we have this idea of junk DNA. And what you see as you study these different groups, as you increase in complexity, that is, as you look at organisms that are more and more complex at the tissue and organ system level, you find greater percentages of the DNA devoted to something other than coding for a protein. Until you get to humans, and you see well over 90% of the human genome devoted to something other than coding for proteins. And so we get this idea of junk DNA. The interesting thing is in 2012 you had a couple dozen papers published all at the same time as part of the ENCODE project that basically illustrated a function for almost all of this DNA, and it's almost entirely regulatory. And so one of these papers noted that there's no section in the human genome that's more than 8,000 bases away from a site of active transcription. Now, not all these sites of transcription are going to be translated into protein. In fact, most of them are never going to be translated. They are transcribed into some type of RNA that is regulatory, that regulates the transcription of other proteins or regulates the translation of RNA into protein or regulates protein function, the processing of the proteins. And so this idea of junk DNA is no longer a real viable idea. And it seems to be that we used to consider all of these examples of duplication events rather are just regulatory genes that are similar in structure to a protein coding gene. And so what looks like it was a duplication event probably is better explained as something that was there along with that original protein coding gene. So now, if natural selection were actually going to produce new structures, if natural selection were going to generate new structures, if this were possible here, where we go from the first protein and we generate an entirely different protein, maybe after some duplication event, if natural selection were going to do that, the question is, what would need to be in place? Well, first, we would need to have these mutations occur exactly when they're needed. If they occur too early, there's natural selection is not going to be able to favor them, and they're just going to get either eliminated by natural selection or eliminated by genetic drift. If they come too late, obviously it's no longer helpful for the organism. Not only do the mutations need to show up when they're needed, but every single mutation along the way, every single one of these mutations illustrated here, say this S changing into something else, into a space here, every single one of these would need to provide some kind of a selective advantage so that natural selection could fix this. Otherwise, there's nothing keeping this from, say, mutating back to this S. And so here in both illustrations, you see this mutation being fixed. And then we've got our next mutation, which is this E turning to an O. And in here, it's able to be fixed because it provides some selective advantage. The reality here is more like this. This second mutation here that goes to a P, I guess this one ends up being fixed as well. But this third mutation here that mutates into what you need, this R here, but rather than becoming fixed as it does in this example here, it actually reverts or goes to something else altogether. And that's more the reality, because the likelihood of you having a single random mutation like this providing some selective advantage so that it can become fixed by natural selection is incredibly low. Really, it's far more likely that it would become fixed completely by accident due to genetic drift. And this would especially be true if the population were small. So natural selection, it can't predict the future, therefore you have to have these mutations showing up exactly when they're needed, and they have to provide a selective advantage. And this is true for every single new protein formed by the organism. And what we just mentioned in the previous slide is the while most of the genome doesn't actually get translated or isn't transcribed into something that gets translated into protein, what you find is that for every new protein you need at least one regulatory element. And so not only is this challenging that every single new protein that you generate is going to require these mutations to show up on time, these mutations to provide some selective advantage every single step along the process of generating a new protein. But on top of that, we're going to need a lot of regulatory material for every single one of those new proteins. So is there something other than the Darwinian mechanism? Because this so far is focusing just on generating these new structures using natural selection generating these new structures. There's some other mechanisms that have been proposed. What about lateral gene transfer? Could something like a virus take a gene for a protein out of one organism and actually insert it into another? We're finding that this happens all the time in bacteria, but we're also starting to see evidence of this happening in eukaryotes as well. finding for example, the gene that codes for latrotoxin, the venom in black widows. We're finding that in other insects, indicating that it was probably transferred that way through lateral gene transfer from a virus. And this is a really interesting idea of how you could generate, macroevolutionary changes by transferring genes. The interesting issue here, though, is that gene had to then evolve in some other organism, right? So sure, that may provide some mechanism by which one organism could change relatively quickly, although we'll talk about some other challenges to that. But we still had to evolve that protein coding gene somewhere. Another mechanism, hybridization. The most famous example of this is endosymbiosis, where you have, say, a prokaryotic cell phagocytize an aerobic bacterium and allow it to live, and that over time becomes the mitochondrion. This same prokaryote could phagocytize a cyanobacterium and over time, or allow it to live, and over time it becomes chloroplast. And then, through time, after this hybridization of these different prokaryotic forms, we can generate something entirely new. We've got the first eukaryotic cell that then can diversify into our various groups of eukaryotes. And this is an interesting idea as well, but it still requires that even if this were true and we had really good solid evidence of this, it still requires each of these organisms developing the specific genes that they have. So again, it just kind of kicks that issue further down the road. The last one is exaptation, and an example of this is feathers. The idea is that feathers evolve first for insulation, and then over time we're able to be modified into flight feathers and used to enable gliding and then ultimately powered flight. And so this idea of an exaptation or a co-option, or sometimes you'll see it referred to as a pre-evolution, is an intriguing idea, but the book presents an issue, and especially as you're going to apply this to, say, proteins, that you have one protein, a simple protein that evolves for, something like metabolizing some new food item, and then now you have this protein in place that could then be co-opted into some other complex molecular machine. The textbook, they address this issue using the analogy of Legos, and they say, okay, well, you've got these Legos that are incredibly well designed to fit together, and proteins that are building these really complex molecular machines with quaternary structure, where you have several proteins built and packed together. They're incredibly sophisticated, far more so than even Legos, but Legos have to be designed with the idea of attaching them together in mind, in the same way that these proteins have been designed with the specific ability to be able to combine together to form this complex molecular machine. And so the idea of actually having this work without somebody with the foresight to match these protein domains is incredibly difficult to imagine how that could actually work. So natural selection and random mutations don't seem to be a good mechanism for explaining how you could get macroevolution. Artificial selection seems to indicate that there's a limit to what microevolution can do. These other mechanisms don't seem to provide a good way to explain how we could generate macroevolution. Well, we have another issue, and the question is, well, if one of these mechanisms could work and could generate new forms, the question is, when would this have had to happen? And what you find is that when you compare all living forms, there's an enormous amount of shared biochemistry among the various phyla of animals, of plants, of fungi, of protists, of bacteria, of archaea. There's just an enormous amount of shared biochemistry. And so what this means is it's pretty reasonable to assume that even the earliest forms in the fossil record, the textbook talks a lot about trilobites, that are some of the earliest animals that appear in the fossil record, it's reasonable to assume that these early animals would have had these really complex biochemical features, these complex molecular machines already in place, which means at the time that we first start seeing a great deal of diversity appearing in the fossil record, all of these molecular machines really need to have already evolved, which means that this thing that's already very difficult to explain mechanistically had to have happened very early in Earth's history. And then we have the question is it even mathematically possible? So assuming we had a good mechanism, assuming we had a good mechanism, is it even mathematically possible to generate these proteins through random mutations? And so the textbook talks about the chance of generating cytochrome C, which is an incredibly small protein, is 1 in 2 times 10 to the 75. And so an astronomically small probability of generating cytochrome C, which is about 100 amino acids, through random mutations, random assortment of amino acids. Now, the average protein is much larger than cytochrome C, and so what we find is that the average protein would require 10 to the 390th power tries to assemble. This is particularly concerning because the approximation for how many events could have occurred in the 15 billion years that the universe may have existed is 10 to the 141 power. And so we have something that's several magnitudes, just astronomically high. And so it would require more tries, way more tries than the universe is even old enough to have allowed. So assuming we had a good mechanism, the probability of even generating one of these proteins is mathematically impossible. When you add to this fact that it seems that every taxon has unique proteins, and some of these taxa may have several thousand genes that are unique to that taxon, then it's not just one protein, it's not the average protein we need to build, but it's several thousand new proteins, new protein coding genes that we have to develop every time we're going to generate a new taxon. And so we have some additional complications. Michael Behe is very famous for his original work, which we'll talk about in a minute, but something he's published more recently I think is really fascinating. He talked about some of these artificial selection experiments. He's talked about the experiments we've done on E. coli and other bacteria where we have studied thousands and tens of thousands of generations, and we see that we just keep bumping up against the edge of what microevolution can do, showing time and time again that the data seem to indicate that macroevolution is not a reasonable extrapolation of microevolution, that there are very definite edges to what you can accomplish. He's most famous for his book on irreducible complexity. He talks a lot about a flagella in that even a relatively simple structure like a flagellum requires, and this is a flagellum of a bacterial cell, requires several different components being in place at the right time in the right place in order for the flagellum to function the way it is meant to function, and that's to propel a cell through some fluid. And so what you have is you have these complex molecular machines that can't be built piecewise, that the only way you could build them is if you could do it through exaptation, where you have all these different proteins carrying out all these other functions, and then you just are able to then build them and compile them all into a single molecular machine. But we've already talked about the challenge of that all of these, in order to fit together, the protein domains have to be perfectly designed in order to fit together and function together. So how is this complicated even further? So Exaptation, we talked about that of the three alternative mechanism we proposed, which was lateral gene transfer, hybridization, and exaptation. Exaptation was, in some ways, I think one of the more intriguing, but we have some complicating issues. So, what you find is there are certain key genes that function very early in development, like here at this stage, that tend to set the body plan for the organism. And so these are the genes that would allow you to generate an enormous amount of morphological change. But what we find is that there are developmental constraints that prohibit those genes from accumulating mutations, at least from accumulating mutations in a meaningful way. So these are really the genes that are acting early in development, that are setting the body plan. Those are the genes you would want to target if you wanted to design a new organism, if you wanted to generate a major branching pattern, if you wanted to accomplish macroevolution. But what you find is those mutations, because of developmental constraints, are always lethal. What we tend to find is mutations that development allows, mutations that aren't blocked by those developmental constraints, are mutations that happen later on in development when the body plan is already set and don't have a significant impact on the overall morphology of the organism. We've talked about epigenetics before, the idea that the environment influences your gene expression a great deal. And so we've talked about how epigenetics could mask mutations. Epigenetics could even influence mutations. And so what this does is it complicates it even further, and it makes it look like that, one, these mutations are not entirely random, that there's a mechanism in place to even to specifically mask mutations if it's to the detriment of the organism, and maybe even to generate mutations needed for the organism to be better equipped to meet its environmental stresses. But something even more difficult with the idea of epigenetics is it seems like virtually all living organisms have the same epigenetic machinery. And so now we have this really sophisticated control mechanism beyond the DNA that has to be evolved and generated incredibly early in the process of the evolution of life if you are going to allow microevolution or macroevolution to happen. And so let's come back to these hypotheses. Remember, the first one is universal common descent is true using the Darwinian mechanism. Next one is universal common descent is true using some mechanism other than the Darwinian mechanism. Next one, an interventionist hypothesis, universal common descent is not true. Macroevolution works primarily by Darwinian mechanisms. And the last one is universal common descent is not true, and even microevolution requires a more sophisticated mechanism, or requires a more sophisticated mechanisms. Gosh, that's two typos because the slide was copied. And so we mentioned this already, but epigenetics and developmental constraints make it very unlikely that random mutations and natural selection could even do a great deal of generating a lot of the microevolutionary changes. So I think hypotheses 1 and 3 look like they're on fairly shaky ground. And at the end of this chapter, when we present these incredible challenges to macroevolution, to me it seems like giving this a very fair treatment, that hypothesis 2 is on fairly shaky ground as well. And so I would be willing to argue that hypothesis 4 not only is the best explanation for the data that we have, but the one that is going to be supported time after time after time as we accumulate more and more data on these areas. So as you watch this presentation, as you work through this, again, keep these hypotheses in mind and be ready to discuss on Monday many of these evidences against macroevolution and what that means.